So we're going to roll with it. Yeah. Okay. Hi, everyone. I hope you guys are doing great. Should be 7 p.m. right now. I do not currently have Wi-Fi, or at 7 I won't have Wi-Fi, so I'm recording this ahead of time, so you guys still get to watch the lecture. Let me share my screen. Uh-oh, full screen. Okay, so here we go. Right. Okay, there we go. Bio week 2. This is your moment now if you haven't gotten water, if you need to go to the bathroom or something like that, because you've had a lot of back-to-back classes. I'll give you about like a minute. So a minute after right now, I'm going to start my class. Okay, in the meanwhile, I'm going to test my speaker. So I have about 20 seconds left. Okay, I don't know why Zoom is saying that, but hopefully you guys are hearing me. Okay, it's been a minute and 5 seconds, gave you 5 extra seconds, and now let's get started. Hopefully you were hydrated. So last week we talked a little bit about the 4 different macromolecules. And we didn't get to fully cover nucleotides because I was missing a few diagrams. So here they are now. If we look at the bottom, this is like the bottom image. This is the main structure of a nucleotide. There are 3 main parts to any nucleotide. And remember, nucleotides are what make up our DNA and our RNA. So we have phosphate, which is on the very left, sugar, which is here, and our base. So this is how we go. So we know if it's adenine versus thymine versus guanine versus cytosine, because if you look in this top right diagram, all of them have a different structure, right? Thymine is different from cytosine, is different from adenine, is different from guanine. And I also talked a little bit about purines versus pyrimidines. And purines are guanine and adenine because you can see that there are 2 rings versus the singular ring in thymine and cytosine. And that's just part of their structure. And so what's cool here is we know DNA versus, okay, so Zoom is covering the bottom diagram, so I can't see if the sugar is ribose or deoxyribose, but basically on the bottom right corner of the pentagon, whether or not there is an oxygen tells you if it is deoxyribose, which would not have an oxygen, versus ribose, which does have an oxygen. And yes, deoxy meaning lacking oxygen. So DNA, sorry, DNA you'll hear if you see in this top right diagram, oops, there is a sugar phosphate backbone, right? You have your bases in the center and the backbone is made of sugar and phosphate. And if you look at this very left, let me see if I can. Nice. Okay, hopefully you can see my laser pointer. If you look at the very sides of this, you see that the phosphate of this nucleotide okay, this is, so we see that the phosphate of guanine is connected to the sugar of thymine, and this phosphate of thymine will be connected to the sugar of something else, and that's kind of how the backbone is built. We also talked about how hydrogen bonds are used to hold the nucleotides together. So fun fact, random fact, you don't need to worry about remembering this, but there are two hydrogen bonds between thymine and adenine, and there are three between guanine and cytosine. So typically the, I guess, chunks or sections of DNA that have more guanine and cytosine are stronger because there's more bonding. And that's always good when you're doing experiments. I guess like for the DNA, if it's in a very heated solution, it is more likely to stay together if it has lots of guanine and cytosine compared to if it was primarily adenine and thymine, because it's easier to break down something that has less bonds. And yeah, I think that is all. The nitrogenous base, remember this can be adenine, thymine, guanine, cytosine, or uracil, again, depending on what the sugar is. And yeah, that is it. Also, DNA is built from the 5' end to the 3' end. So you can see here, there's a 5', and we go to a 3', right? And here's 3, which goes to 5. Basically, 5 is this like phosphate area, 5', 3' is this OH group. So 5 to 3, you're always adding phosphates to that sugar. So you're building on the 3' end. If that didn't make too much sense, it's okay. We went over DNA replication last class, and that's just used there. Okay, now people for some reason had interest in cellular respiration. So we'll be going over cellular respiration. This might be a familiar term to a lot of you guys, but you may not know what exactly cellular respiration is or does. So Google's definition of cellular respiration is the process by which organisms use oxygen to break down food molecules to get chemical energy for cell functions. And we always know like we need glucose, or you should know that glucose is very needed for survival. It is important for your energy, and it's important because cellular respiration uses glucose. And the ultimate product, or like the reason we would need cellular respiration, is because ATP is produced. And ATP is our primary form of energy. Any process we do that needs energy, anything that needs to be broken down or transported or created, will use ATP. And ATP is adenosine triphosphate, just FYI. And most of the steps of cellular respiration, I guess technically all of the steps happen in the mitochondria, which is again like tying back to why we call it the powerhouse of the cell, because all of the energy producing steps that our body does is done in the mitochondria. So basically cellular respiration is done in the mitochondria. However, there is a first step, which is called glycolysis, that does happen in the cytosol. So you can see here glycolysis. You can assume that anything white is the cytoplasm. So glycolysis will create pyruvate, which we'll go over in a little bit. And then everything after that happens in the mitochondria. So that's why it's kind of like iffy. Some places consider glycolysis to not be a step in cellular respiration. I would say it is a step, just because if glycolysis doesn't happen, nothing will happen. So it should count as a step. And so yeah, the main steps of cellular respiration starts with glycolysis, which I talked about just now, happens in the cytoplasm. Then we move on to pyruvate oxidation. Then it is the citric acid or Krebs cycle. And lastly, we have oxidative phosphorylation. Now, I wouldn't try, if this is your first time learning about cellular respiration or any of these steps, I wouldn't try to memorize everything like right now, because then you'll get confused with all of the steps. So what I did for this class is it is just a very, very nice general overview of everything. And you can just kind of absorb it. If you plan on taking notes, I would say maybe bullet out the different points. So bullet out glycolysis. And right now just learn, listen, absorb everything. And then you can go back and go, okay, what did I take away? And do memory recall, as they say in psychology, and see whatever you could remember from this class. If we just look at this general diagram, Oh yeah, it's labeled cytosol and mitochondria. Here we see glycolysis actually makes 2 ATPs. Citric acid cycle makes 2 ATPs, but the main source of energy created, or ATP creation, is from the oxidative phosphorylation because there is an electron transport chain. And we'll look into that. So first, we have, oh no, it's not switching slides. Hello. Oh, there. Glycolysis. Glycolysis, like I said, it happens in the cytosol. Glycolysis in whatever language, I would assume like Latin, maybe not, means sugar splitting. And like we said, you need glucose, right? Sorry, I don't know if I mentioned this. You need glucose for any of this to happen. Glucose is our source of energy. So in glycolysis, we're going to end up, the main job is to convert glucose to pyruvate. Now, what are both of those? Glucose, we talked about this last class. Glucose is a sugar, and its chemical formula is C6H12O6. And how you understand that is that there are 6 carbons, 12 hydrogens, and 6 oxygens. Key takeaway, there are 6 carbons. And whenever you break it into 2 pyruvates, the only thing that you need to remember about their molecular structure is that each pyruvate will have 3 carbons. And also, obviously it's not just glucose is converted to 2 pyruvates and that is it. There is also water that's created, hydrogen is created. There are 2 ATPs and 2 NADHs created here. NADH is important because what's going to happen is all of these steps, for the most part, are creating a lot of NADH. And during this last oxidative phosphorylation electron transport section, these NADHs are going to be, the energy within them are going to be used to create ATP. So you can imagine it to be like pre-ATP. It's basically ATP. It's not ATP. Okay, it stores a lot of energy, and that energy can be used to create ATP. And we like ATP because ATP is our source of energy. So yeah, that is glycolysis. Key takeaways are that glucose is broken into pyruvate, and you're making 2 ATPs and 2 NADHs in this process. Remember that there are also other byproducts like water and hydrogen that are made, but I just wanted this class to be like very gist of everything, and if you remember this, you basically know it. Glycolysis does have like 12, 20, a lot. There are a lot of sub-steps, and it goes over the very specific molecular function. But not only would that be a very difficult class to follow, it would be very boring, and I don't know if it's worth it to memorize all of those steps. So I think it's always good to just know what's going on, and you can apply it later on. Next, we have pyruvate oxidation. So if we go back to that first diagram, you can notice that glycolysis is happening in the cytosol, but pyruvate oxidation is the first step that happens in the mitochondria. Now, so you have this pyruvate here that was made from your glucose. It will enter the mitochondria through a transport protein, and I'm not going to go into the different types of transport proteins, but there are many different types, many different functions for these proteins, and they basically help molecules get in or out of the cell. Yeah, that's about it. No need to worry too much for this. But yeah, pyruvate, you can now know, is inside of the mitochondria. Bye-bye cytosol. And there are 3 main things that happen to this pyruvate molecule to get our final goal, which is acetyl-CoA. So glycolysis, we converted glucose to pyruvate. Pyruvate oxidation, we're going to convert pyruvate to acetyl-CoA. Did I say that right? Glucose to pyruvate, pyruvate to acetyl-CoA. And first thing that's going to happen, pyruvate's transported to the mitochondria, and then, yeah. So first, the COO minus group that you can see in the blue, that's removed. So the first thing that happens is we kick it out. Goodbye. And that is released as a CO2, which carbon dioxide, it is a waste product. You should know what it is. That's the first thing that's done. We remove that. Second is you have the remaining section of that pyruvate oxidized, which is why this step is called pyruvate oxidation. And basically, oxidation is the removal of an electron. So this blue guy has an electron, and then it's oxidized, and it doesn't have the electron anymore. It lost it. Versus if you ever hear anything is reduced or there's a reduction, you have this red molecule, and it has an electron, so it gained it. Reduction is gaining an electron. Oxidation is losing an electron. I like to remember it by charge. So when you're reducing your charge, you're more negative. Electrons are negative. That might be confusing. I know there is an acronym that some people use called oil rig. I don't know what it stands for, but I know it helps with this. So if you are very curious about redox reactions, probably search up oil rig. Oh yeah, oxidation is oxidation is loss. Reduction is gaining. Probably something like that. But that is all that happens. So if our pyruvate is oxidized, it's losing the electrons, right? And these electrons are going to be used when converting this or these electrons are going to be used to convert NAD+, which is in the mitochondria, into NADH plus hydrogen. So this is a simple chemical reaction, but you can see that we're stocking up on this NADH, so we can use it later on. Because if we were to we can't make ATP directly from this. What we can do is create a molecule that stores a lot of energy, is high in energy, and later on break that down and use that energy while it's breaking down to make ATP. So that's what we're doing here. I actually think this step, so pyruvate oxidation makes no ATP, but it does make NADH, which is great. So pyruvate is oxidized, and then CoA is added, right? CoA is coenzyme A. Just know that it's added to the pyruvate molecule, what's left of it, and there is also a sulfur. A lot of places note that there's sulfur in this, so I will tell that to you as well. You have a sulfur and your coenzyme A, or it's part of your coenzyme A. Okay, next, if I'm going too fast, I might be going too fast. After I go through this, I'll rerun through everything once before we move on to the next topic. I also, you might have noticed as we were going through the steps, I mentioned briefly of how it's going to be used in oxidative phosphorylation, and I'll go over oxidative phosphorylation in the next class. I don't want this to be too overwhelming with all of the steps thrown at you at once, so we'll go through the first three and then the last one next Friday, because that stuff is a little more complicated than the other ones. So yes, citric acid cycle. Again, do not worry about memorizing all of these different molecules and all of their chemical formulas. This is like, I think every step of cellular respiration, you could find something that has it, unless you are being like directly tested on what is like the third subproduct, byproduct, molecule formed. Don't worry about this unless it helps you remember it, which I don't know why it would. But so here we have step number 1. You have your acetyl-CoA, and it's converted to citrate. Again, don't worry too much about these names. But what we want to take away from this citric acid cycle is you can see the things in orange are good things. We like these things, and they're produced every time, every cycle. So you have 3 NADHs that are made here. You also have an FADH2. This molecule is, you can imagine it to be synonymous to NADH. They both store energy, and they're going to be used in the last step to create more ATPs. They're obviously not the same thing. That's why they have different formulas, but they basically do the same function. So if you're not too worried about like specifics of molecular structure, you can remember it like that. There's also a lot of CO2s, not a lot, I think 2, that are created. So CO2 here, CO2 here. And that's the main takeaway that you should take. There are 3 NADHs, 1 FADH2, and an A2P that are made. And that's very beneficial. That's a lot of energy. So you know that 1 glucose makes 2 pyruvates, each of which are converted into an acetyl-CoA. So now we have 2 acetyl-CoAs per glucose. And so every glucose that is made, this cycle will happen, or every glucose used, the cycle will happen twice, which means you'll end up with 6 NADHs, 2 FADH2s, and 2 ATPs just from the citric acid cycle of 1 glucose molecule. Yeah, this cycle is interesting. It talks well about, learning the molecular mechanisms behind this is not harmful. It's interesting to see like how one thing is changed into another, but don't stress yourself on memorizing it. So I'll touch into this, but again, don't worry too much. This fun stuff. So yeah, we have our acetyl-CoA, and it's converted to citrate because you get rid of this CoA SH section. And then citrate is converted into isocitrate by removing a water molecule. So if we look at the structure, okay, we see there was a hydrogen lost here, an oxygen here. We need another hydrogen. Maybe this, okay. So we just know citrate loses the water. And then isocitrate is oxidized. And if you remember what oxidized means, it is when you lose an electron. So NAD plus is converted to NADH because the electron is lost. And this, whatever is left of this, loses a CO2. Then you have alpha ketoglutarate. So another carbon dioxide is lost, and whatever is left is oxidized. So whenever you have something that's oxidized, something is typically reduced at the same time. And that's why it's called redox reactions, reduction, oxidation, right? If something loses an electron, something has to gain that as well. So in this case, when this molecule is oxidized, NAD plus is reduced to NADH. And every time that happens, it's good for us because we want to stock up on those NADHs. Yeah, I don't think you need to know. Don't worry about this. I don't want to overwhelm you guys, and I don't think it's needed. So, like I said, next class we'll go over oxidative phosphorylation in depth. Brace yourselves, get ready for this. But in the meanwhile, let's go over these steps one more time, and then we'll move on to cellular division. So cellular respiration has 4 main steps. We have glycolysis, pyruvate oxidation, citric acid cycle, and oxidative phosphorylation. Glycolysis is when we convert glucose to pyruvate, and we end up making 2 ATPs in this process. We also make NADHs, which is great. Then your pyruvate is transported into the mitochondria, loses a carbon dioxide, is oxidized, adds a sulfur coenzyme A, and now we call it acetyl-CoA. Then this acetyl-CoA enters the Krebs cycle or the citric acid cycle. If you're torn over which name to call it, I think it's always safe to say the Krebs cycle. Maybe. It doesn't really matter. And you have this acetyl-CoA that is converted into, well, your takeaway is 3 NADHs, an FADH2, an ATP, 2 ATPs. Yeah, and then those NADHs and FADH2s that we've been stocking up on, is going to enter the oxidative phosphorylation phase, section. Yeah. Now we should be at the 30-minute mark. Whoops. And now we can talk about cellular division. This image, I don't really know what it is. PowerPoint suggested to add it and it looks cool. So we have that. And cellular division is basically, what you can imagine is you have your cell. Your cell needs to divide. So what it's going to do is it's going to double the amount of cytoplasm. It's going to double the amount of organelles and then essentially like break in half. However, if you remember, our cell has a singular nucleus, right? And both of our cells need that nucleus with all of the chromosomes or else they're not going to function properly. So the really like relevant part of cell division is mitosis, or that division of the nucleus and chromosomes so that each cell gets the right number of chromosomes. So interphase, so although mitosis is, like I said, or I guess it's opinionated, but it really is the most important part of this. It is very short. So if you look at this diagram, this is like cell division across time. So the first, and the like, so this is like a pie chart. And if you know how a pie chart works, like the bigger it is, I guess like the more percentage it is. So G1 is a long phase. It takes up a good amount of the circle. So interphase is divided into G1, S, and G2. Those are the three subsections of interphase. I'm not going to go, oops, no, shaking. I'm not going to go into them because I don't think anything ever talks about them. You just need to know everything's dividing. DNA synthesis isn't important. It is important, but we already talked about DNA replication before, so it's just DNA replication. Yeah, so we have G1, S, G2. This takes up most of the cell cycle. For like if you're to pick a random cell, odds are it is an interphase because this is like what, 90%, I think, of the time that the cell spends as an interphase. And then there is a small mitosis, which is the division of the nucleus. Hello? Okay. So yes, now we'll go on to mitosis. What's really nice about mitosis is once you read it 2 or 3 times, you can look at this diagram and go, yep, I know everything. And the goal is for you to know everything, right? So these are all of the steps of mitosis. Don't be overwhelmed by this. And I did say, you know, some parts of cellular respiration, it's not important to memorize like everything. I would say here, try your best to memorize steps and just like a little bit of what happens in each of them. If you are familiar with mitosis, something that may not be so familiar to you is prometaphase. Some textbooks will say it goes straight from prophase to metaphase. Others will say that there is an in-between phase called prometaphase. So I think it's always safe to learn prometaphase, and then if something is not asking for it, you can decide whether that function or process is more relative to prophase or metaphase. So mitosis steps, we're starting with prophase. Prophase is like basically the nucleus and your chromosomes inside of the nucleus are getting ready to divide, but nothing is really happening at all. So in a normal cell, can I zoom in? I have no idea if this is zooming in. Okay, escape. Basically, oh, there we go. Okay, this is better. My laser pointer is gone. No. Technical difficulties. Okay. Basically, pay attention to this nucleus on the top left. You can kind of see these like little squiggly things here. And essentially, while your cell is in interphase, which is most of the time, your chromosomes are not going to be those compact things that you're typically accustomed to when you look at a chromosome. They're going to be really sprawled out, really wide. They won't look like a chromosome at all. Under a microscope, it'll just be a bunch of blobs, not blobs, spread out, crazy shaped things. And that is because you want your enzymes to be able to access the DNA, right? And if it's so compact and so condensed, it is very difficult for you to be able to do anything with it. And we talked about transcription, which was the process of converting DNA to RNA, which later becomes protein. So say your body is like very low on insulin, and you want to create that protein, right? So you would have to transcribe your DNA to RNA to protein so that you have more insulin. And the only way you're able to do that is by accessing those chromosomes, which encode for how you make certain proteins, which is why they're normally very loose. But whenever they're dividing, they become condensed, compact structures, and that's what you see as a chromosome. So these X's are those compact chromosomes, each X. Oh, by the way, humans have 23 pairs of chromosomes, so total of 46. 44 of those are somatic, the right word, autosomal. Just chromosomes that are in everyone. They're in everyone, right? And the last two are either XX or XY. If you have two X chromosomes, you are female. If you have an X chromosome and a Y chromosome, you are male. For your birth, that's how you are birthed, your gender when you're born. Yeah. So we have prophase, which is preparing to divide, and you want to condense those chromosomes because, I mean, if you think of it logistically, which is not maybe the best way to think about it, but it will make more sense. If you have a bunch of sprawled out things and you're trying to separate every single piece, it's very time consuming, it's very difficult. And you want this process to be spot on because these are your chromosomes, right? Typically your cellular machinery does not mess up. But if it does, that's really bad. We don't want it to mess up. So when you're having a lot of sprawled out, confusing stuff, it is difficult to divide it, right? In comparison to if you condense everything and you try to divide these really nice, smaller things, it works out well. I'm trying to think of a analogy. I guess you could a slinky, if you guys are old enough to know what a slinky is. I don't think those are very used anymore, but it's this spring that you can stretch out really wide, and you also can condense it. So you can imagine you have a bunch of slinkies that are stretched out. That's going to be very difficult to work around. They're going to get tangled and they're going to be so gross and they'll hit your fingers, versus if you condense them into those small cylinders that they can be, it's really easy to stack it and sort it, right? And that's kind of the goal. We want our chromosomes to condense, and in this process, they also become visible. So when you look at it under a microscope, you're going to see these X shapes that we're accustomed to chromosomes being. Then I told you guys last class we'll start talking about spindle fibers and centrosomes and centromeres. So this is where they're applied. You have spindle fibers, which are these blue things that come from each end of your cell. From each end of your cell, and they're used to pull your chromosomes apart. So basically, you see these X's here. You have 3 X's, right? And these are, I guess this cell has 3 chromosomes. What happens is you take 1/2. So every chromosome here is made of 2 chromatids. Let's see if I can. Okay, let me draw. This is not a good page to draw on. Let's go back. This is a nice clean slate. Okay, we have chromosome. This is a chromosome. Oops. And you may have noticed, you should have noticed, you have these two lines that I connected together with this, like, I guess you could pretend it's a protein, right? But each of these lines are a chrome. That was such an awful D. These are all chromatids. So you have two chromatids that combine together to make a chromosome. And during mitosis, also these chromatids are like basically the exact same. This will code for protein 1. Oh, this is so awful. Just here, I'm not going to draw anymore. This codes for protein 1, this codes for protein 1, this codes for protein 2, this codes for protein 2. So they do the same thing. And what happens in mitosis is this will divide into these two sections. So one cell will get this, one cell will get that, and we'll all live happily ever after because all of your cells, your two daughter cells that are made, will have their DNA. So that is what is happening. Your spindle fibers are the reason you can detach your chromosomes. Nuclear envelope. So we know that there's a nucleus. You may know that there is an envelope surrounding it. Just you can imagine the cell membrane. It starts to break down. Why does it break down? Because you can notice the spindle fibers are on the outside of the cell. They're not inside of the nucleus. So in order to access it, you need to break those down, right? And it's also another reason the nuclear envelope breaks down is because you're going from one nucleus to two, and it can't just like pinch itself in half. So what is going to happen is like you'll remove the whole nuclear envelope, and then you'll end up creating two more, which you can see in the telophase section. And then yeah, your centrosomes. So these things start, the red blue things start to move towards separate ends of the poles. Why is this? Because again, we're pulling the chromosomes in different directions. And that's how you can imagine like if they were both on the same side, you're pulling the same thing toward it wouldn't work. It wouldn't break apart, right? And that's the goal. So that's what happens in prophase. You're just kind of preparing for chromosomal division, mitosis. Yes. Prometaphase. So metaphase is basically, if you're going to know anything about metaphase, is your chromosomes are lined up. You can see here there's a nice line of chromosomes. They're all aligned. They're attached to their spindle fibers. Life is good. Prometaphase is like the process of slowly having those chromosomes align to the perfect shape, state of being that they're in metaphase. So in prometaphase, your chromosomes are going to keep on condensing to get into that like prime form of greatness, and kinetochores will appear. Don't worry too much about that. And mitotic spindle microtubules attached to kinetochores. So just these blue lines are attaching to your chromosomes. That's all you need to know. And by metaphase, all of them are attached, all of them are lined up, everyone is thriving. Oh, wait, I didn't. Let's look at these bottom images too. These are real cells, obviously under a microscope. They're illuminating. It looks really cool. So here we kind of see there's like a jumbo mess of stuff. These green things are those mitotic spindle microtubules. Microtubules are also made of a protein called tubulin, just like in the name. So it's really easy to remember that. So you have these green lines that are your microtubules, and notice how like the blue from here is very different from the blue from here. I'm assuming those blues are the blue is the chromosomes. So you can kind of see like how everything is condensing to this state here. And yeah, nice stuff. Next, we have anaphase. Anaphase, the key takeaway is your chromosomes are divided. You have detached them, you have detached the two, sister chromatids. Now each chromatid is called a chromosome. So the naming behind this is really weird. It took me very long to figure out like what is happening. But basically, every chromosome here has two sister chromatids, right? And we are dividing it. So now we have, you would say, 6 sister chromatids, right? Wrong. This is called 6 chromosomes. So yeah, I don't know why it's like that. But once you get it, you get it. So just know that each of these are now called their own chromosome. So you have 3 chromosomes going to the right end, 3 chromosomes going towards the left end. Good stuff. So yeah, centromere split in 2, chromatids, yeah, sister chromatids that are now called chromosomes are pulled towards opposite poles, and the spindle fibers begin to elongate the cell. So the ultimate goal of cell division is so that you have one cell that is now 2 cells. Again, if we look at logistics, it is a lot harder to split this in half, prometaphase, than to split anaphase in half. See how clear this like line of division is? It makes life a lot better when it's a longer cell. But again, this is a diagram. Like I have full 100% doubt that, you know, all of the organelles have moved to their respective sides. There will be stuff here in the middle, and that's how life works. Your cells will still be fine. Yeah, it's okay. But it's not this like clear cut distinction that they're showing, which is just there to make understanding a little bit easier. And then lastly, we have telophase. Some people say telophase. I think it's telophase. It sounds better as telophase. Oh yeah, if I've like mispronounced anything in the past, just like pretend I didn't do that. I haven't like spoken these terms out loud in a while because like who am I going to talk to about telophase or cellular respiration? Especially because COVID. So just pardon me on that. Should be okay as long as you know what's happening. So yeah, telophase is when chromosomes arrive at opposite poles and they're decondensing. So now they're going back to that form that the chromosomes were originally in, which was that loose shape, state of being. And then the nuclear envelope material. Yeah, so now you have your nuclear envelope that is reforming, and you have 2 of them because ultimately this will become 2 cells, right? Your mitotic spindle breaks down. Why? Because you don't need it anymore. You're not dividing, you're not pulling on any chromosomes, so goodbye, right? It's done its function. And yes, that is all. Then lastly, cytokinesis. I don't know if this was like a lot of, okay, when I originally learned about mitosis, which was like years ago, cytokinesis was always associated with telophase. It would be like your phases are prophase, metaphase, anaphase, telophase, and cytokinesis, but it was like telophase and cytokinesis. Those are grouped together. I don't know if it counts as an actual like phase of mitosis, but basically all that it is the cell membrane like what am I doing? Okay, you have your thing and it like pinches inwards. So now you can have 2 cells. That is what cytokinesis is. It is different in plant cells and animal cells. What I just described to you was in animal cells. It's called a cleavage furrow. And it just like splits off. But plant cells, we talked about how plant cells also have a cell wall, and that makes things more complicated. So you have a cell plate that is used, and it's like it's like a plate, clear cut, that is like here, and then your cells divide, from what I understand. But again, this is not plant class. Plants are interesting, but not as cool as humans or animals. Doesn't really matter too much. So yeah, that is all of mitosis. I would suggest like maybe not today because it would be what 8 p.m. by the time this is done. Maybe tomorrow morning. Don't ruin your Saturday, but if you have time, like come back and look at this. You can easily Google like phases of mitosis and just make sure like you can recall this because I think it's always good to know. Like this is very simple genetics, simple cell stuff, and it's great to know this. So try to look back on it if you can. And next class, I'll probably have like a small recap anyways, um, just so you have like this continuous reinforcement of what mitosis is. So one last time, let us quickly go over these phases and I'll tell you like if there's one thing to remember this, right? Prophase is your chromosomes are condensing and your nuclear envelope breaks down. Those are your two like important. Oh, let me see if I can highlight on this. I think I can. Let's see. Oh yeah, there we go. Okay, chromosomes are condensing and the nuclear envelope breaks down. Maybe at the end you can take a photo of the screen or just write it down as I go. I'll start talking a little slower so you have a little time. So prophase, chromosomes condense, nuclear envelope breaks down. I'm not going to go over prometaphase because it is basically just like prophase becoming metaphase. Just continuation of prophase, really. Metaphase, you have your chromosomes. Wow, that was so ugly. Okay. Your chromosomes are lined up. Okay, that is all you need to know for metaphase. Your chromosomes have lined up. Oops. This is like, Zoom is not being kind. There we go. Chromosomes are lined up. Now we have anaphase, which is your sister chromatids pulled towards opposite poles. That is what happens in anaphase. You're dividing your chromosomes into new chromosomes because naming is weird, but that's just how it is. And then telophase is when you basically, it's just like you're basically, telophase is you're done doing the actual division and now you're like fixing up for the cell to become two, or your nuclear division is done. So we could say chromosomes arrive at opposite poles. And you can also note that nuclear envelope forms. Oh, that's so horrible. Oh, I can type? Nice. Forms. Okay. You have nuclear envelope forms. Oops. Okay, that says forms. And then lastly, cytokinesis is just, remember cleavage furrow. Your cell does the final splitting and it is now two. And now you know mitosis. Sorry. It's 7 in the morning now. Which is why my voice is kind of gross. Yes, mitosis is done. Cellular respiration is almost done. We are doing fantastic. If you have any questions, maybe you have them written down so you can tell me, or you can always email me. I gave you guys my email last class. On my clock, I should have started at 7 a.m. and my phone says 7:55. 5 minutes of pondering your questions, writing them down somewhere so I can come back to them. Yeah. I know someone last class asked me about if we could integrate like jellyfish DNA and expand our lives, and we were going to talk about telomeres, but I did not have time to add that to this lesson. So maybe I will do it next time. If not, I will send you a personal email explaining to you my findings. And that is all that I have for this class. You guys have, 7:55. You're good to go. You can leave the Zoom now, and we'll pick back up from oxidative hello.